Hello, I'm Dr. Charles Knight, and this is my 7th installment in my 10 lectures on evolution channel on YouTube. And welcome back. There's 6 other presentations and at least 3 more to follow after this. I'm from Cal Poly in San Luis Obispo, California. I work in the Bailey College of Science and Mathematics in the Department of Biological Sciences, and this series of lectures is for my Bio 414 evolution class, and hope you enjoy. The paper we'll discuss today, or I'll discuss today, is titled EMX2 Underlies the Development and Evolution of Marsupial Gliding Membranes. It was published on 2nd of May 2024, last year, and it's a great story about evo-devo that I've chosen to bring to my evolution class. It was produced by a whole series of investigators. Clearly, Ricardo Mallarino probably was the lead PI on this paper, and Jorge Marino was a graduate student who completed this work and is the lead author. But there's so many other investigators involved here. I feel always remiss not pointing out more of them, but I want to go into a little bit of detail on these 2 investigators in a scientific vignette on the next slide. But first of all, this paper is about sugar gliders and about early development of these sugar gliders and the Patagium, which is the gliding membrane in these marsupial gliding animals. So Ricardo Mallarino is a professor at Princeton University now. He got his start at the Universidad de Los Andes in Colombia. That's where he got his Bachelor of Science degree, and then went to Harvard from there and worked with Arhat Abanazov on the morphology of beaks. And it seems to me, looking at Ricardo's CV, that his major focus, or a major focus of his work, has been on non-model systems. That impetus probably started with Arhat's work and his work on the Galapagos finches. I'm laughing to myself because I'm saying that they're non-model systems. Well, sure, it's not the chicken, but of course, these Darwin's finches, Galapagos finches, have been studied a lot now, but really Arhat and Ricardo have opened up that field of developmental biology research to this group of organisms and many, many others as well. He continued that work with a postdoc with Hopi Hoekstra on the African striped mouse, I think it is. It's not a skunk, but it's a striped mouse, another non-model system. But he went from working with Arhat at Harvard to then doing a postdoc with Hopi Hoekstra, also at Harvard, working on another non-model system. And then, of course, today's paper deals with another non-model system, which would be the sugar gliders, or just gliding marsupials in general. It's not just the sugar glider that they worked on. And then I'd like to talk a little bit about Jorge Marino, who is from Iowa, of all places. Well, I mean, everybody's from somewhere. He's from Iowa. He got his BS at the University of Iowa, studying the genetic architecture of these little snails called the New Zealand mud snail. It looks big there, but they're actually very tiny. These are a bunch of them around a US dime. They're invasive in the US, and I assume there was money to study these snails because of the invasive aspect of them. They've invaded, when I read Jorge's biography, I saw that they've invaded all the way up to Yellowstone and the beautiful rivers there. So it's pervasive across the entire West, and I'm not sure what the current status is in mitigation or what the future is, because they're everywhere now. But Jorge has moved on. He went to Princeton from the University of Iowa, and it has by now, I'm sure, received his PhD, working with Ricardo Mallarino on these sugar gliders. And what a paper they've produced. It's an amazing body of work that they've produced that brings to light a whole new a bunch of new genomic techniques, things that are new to me It was a challenge for me to understand all of the techniques that were used in this paper, and I'm not going to do them justice here, but I'm going to try at least brush across the surface of some of them so that we can understand what they found. But it's an impressive body of work. So in producing this presentation, a news article that was accompanying this article also helped a lot. It was titled "How a Skinfold for Gliding Emerged in Marsupials," and it was by Dario Lupianez, and it was so useful for me as I'm a botanist. So as I'm developing this presentation, it's nice to have kind of a generalist guide to this really dense paper that produces some incredible new results in evolution. Also, I'm using this evolutionary analysis book. This is our textbook that we use for the class. There'll be some textbook tie-ins, but not too many. And then, of course, I just like to say I use Wikipedia and Google as well. Google a little bit less since the AI revolution there. It's been lots of hallucinations there, but still sometimes have to search for techniques like micro-C and other new things to try to understand what they are. So I always start these presentations with a series of take-home points that are kind of 5 things that I think we can conclude are important take-homes. And I'll start with a simple one, that sugar glider Patagium, which are the gliding membranes, have evolved repeatedly in marsupials. And I was just talking to somebody in the hallway saying that there's a convergence of convergences where there's convergent evolution of marsupial flying behavior. There's placental mammals like the flying squirrel in the Pacific Northwest that fly, but then also the sugar gliders, which are marsupials. There's a convergence there, and that's a kind of a textbook example of convergence. But then within the marsupials, there's been repeated evolution of this flying behavior and morphology. So there's been a convergence of this convergent kind of morphology, which I think is pretty cool. And today we're going to delve into the genetic aspect of what underlies that convergence. So this is a dense take-home point. Transcriptionally active genes in the Patagium were discovered and compared to glider and non-glider genomes to identify glider-associated genes with elevated substitution in cis-regulatory elements. I'm laughing to myself because it's a dense statement. But at the end, you see what the goal here is to identify cis-regulatory elements that have elevated levels of substitution that are in gliders compared to non-gliders. But first they had to find these cis-regulatory elements that involved a lot of new techniques at looking at chromatin accessibility, transcriptionally active genes, and then comparing lots of genomes and sequencing genomes. They sequenced 15 genomes in this study. Brand new genomes to science, had not been sequenced before, non-model species. Kudos to them for doing all that work. But it's also interesting that the end goal, or at least the discussion, revolves around the regulatory regions in this paper, not the protein sequences. We're so used to looking at a paper like this that identifies a gene like EMX2 and expecting to hear about an amino acid substitution in that gene that leads to a different function. Here, that's not the story. It has to do with regulation, and their main point is there's different paths to this regulation, but EMX2 seems to be involved in each case. Each time the marsupial flying behavior has evolved, behavior and morphology has evolved, EMX2 is associated, but the regulatory mutations may be somewhat different in each lineage that evolves this morphology. So they're looking for increased substitution in these cis-regulatory elements around genes associated with the gliding behavior. EMX2, again, stood out in their transcriptomic, epigenomic, and genomic pipeline. They really built a pipeline, bioinformatic pipeline that sifted through all of the genes. This was not a candidate gene study. It was agnostic to the genes, and EMX2 stood out in the end as a gene of major interest. And then they did a series of incredible far out biotechnology techniques to transform joeys in the pouch to underexpress EMX2. So they did a technique like RNAi to underexpress EMX2, and then they overexpressed EMX2 in transgenic mice. And in both cases, they showed that there was an altered Patagium primordia, or lateral skin in the case of mice, showing that this gene does have an effect on that area of skin in these animals. And lastly, I think one of their main points is that gliders show lineage-specific regulatory evolution towards this altered EMX2 expression. It's not the same regulatory elements, it's often different regulatory elements that lead to overexpression of EMX2 or altered expression of EMX2. All right, so I always read the abstract, and this one's fairly long, but I like to read it because all of the authors on this paper put a lot of effort into crafting this abstract into a dense, information-rich introduction to their paper, and I think this is the best place to start, so I'll start with it here. I'll read it slow and try not to elaborate much as we go. I'll save that for later. Phenotypic variation among species is a product of evolutionary changes to developmental programs. However, how these changes generate novel morphological traits remains largely unclear. Here, we studied the genomic and developmental basis of the mammalian gliding membrane, or Patagium, an adaptive trait that has repeatedly evolved in different lineages, including in closely related marsupial species. Through comparative genomic analysis of 15 marsupial genomes, both from gliding and non-gliding species, we found that the EMX2 locus experienced lineage-specific patterns of accelerated cis-regulatory evolution in gliding species. By combining epigenomics, transcriptomics, and in-pouch marsupial transgenics, we showed that EMX2 is a critical upstream regulator of Patagium development. Moreover, we identified different cis-regulatory elements that may be responsible for driving increased EMX2 expression levels in gliding species. Lastly, using mouse functional experiments, we find evidence that EMX2 expression patterns in gliders may have been modified from a pre-existing program found in all mammals. Together, our results suggest that Patagia repeatedly originated through a process of convergent genomic evolution, whereby regulation of EMX2 was altered by distant cis-regulatory elements in independently evolved species. Thus, different regulatory elements targeting the same key developmental gene may constitute an effective strategy by which natural selection has harnessed regulatory evolution in marsupial genomes to generate phenotypic novelty. It's a dense abstract, but they have a lot to say, and they have a lot of impressive results to show. So I always kind of circle some of the words that I think it's important for my students to know, and then some other phrases that kind of underlie what they found. And so I'll go into those words in the next slide, but then just the remarkable fact that they identified cis-regulatory elements that may be responsible, not an altered gene, but regulatory elements was the goal here. And I really feel like this is the next step in developmental evo-devo work. Sean Carroll talks about it a lot in his book, Endless Forms Most Beautiful, right there, about how regulatory evolution is so important. But it seems to me in the last 20 years since the publication of Endless Forms Most Beautiful, it's been fairly slow, the study of the elements themselves. I have a feeling that it's only been really in the last 5 years that the techniques for chromatin accessibility and the interaction between domains has been really become more tractable to study, and this is one of those papers that is at the cutting edge of doing this evo-devo work at the level of regulation instead of just the sequence Notice I just adjust the sequence of the proteins. That's so 1990s. Now we're beyond that. We're at the regulation level, which is great to see. Another statement may have been modified from a pre-existing program found in all mammals. So they study this in sugar gliders, and then they apply it to mice, and they see something similar to the development of the Patagia, which is cool. And also separate paths towards the evolution of this Patagia. We've been dwelling on the statement from Ernst Mayr about how a statement about how all roads lead to Rome is true in evolution as in daily affairs. I think that's something that at least Sean Carroll says that Ernst Mayr said. And the idea that there's a single road that leads to a morphology is controversial, and we're seeing here that there's kind of both here. There's one gene, EMX2, that seems like it's important, but there's separate paths to the upregulation of that gene. So it's not a single road, but it does have a constriction point in a way. What I told my students in class is that the fact that this paper has a single gene in the title, I always say I speak with my breath and my pauses, because often I'm trying to get students to realize that it's the genomic architecture of the phenotype, and it rarely has to do with a single gene, but often there is a gene with a significant effect, and I try to get away from the gene for arguments and think more about the genomic architecture. Here we're really talking about the regulation of this gene, and I'm sure Ricardo and Jorge would agree that there's other genes that are important for this phenotype as well. But this one is a new gene that they've discovered is important, clearly. All right, so I go to these glossary notes slides where I just present some of the important words. Patagium is the mammalian gliding membrane, and I guess that's true for flying squirrels as well as marsupials. So flying squirrel would have a patagium as well as a marsupial. I might be wrong there. I'm a botanist again. Cis-regulatory regions are regions of non-coding DNA which regulate the transcription of neighboring genes. In this study, we're going to be looking at elevated rates of substitution in those regions, which is analogous to positive selection, but we're not talking about non-synonymous mutations because it's just a substitution in a binding sequence of an enhancer or a signature sequence. And we're calling those cis-regulatory elements, those elements of DNA that are close by to the gene that are regulating expression of that gene. But any substitution could lead to altered binding with a transcription factor, not just there is no such thing as a non-synonymous cis-regulatory mutation. Maybe a loss of function, loss of binding type mutation. That's in contrast to trans-regulatory or mutations, which are regulation from a different location or a different molecule. And epigenomics is the complete set of epigenetic modifications on the genetic material of a cell. I don't know if that's a very good definition, but in this case, we're going to study the chromatin accessibility, which certainly is due to epigenetic modifications, and the DNA has to be accessible for expression to take place. So really, this study started with looking at what regions of DNA are accessible in glider species, and of those regions, which ones are cis-regulatory elements, and kind of in the pipeline, narrowing it down to genes. Transcriptomics was study is the study of the DNA that's transcribed to RNA. So RNA-seq is a way to do that. And then in-pouch marsupial transgenics is, in this case, they used the lentavirus that expressed a small little piece of EMX2, which behaved as an RNA interference and decreasing the EMX2 expression in developing joeys, and they saw the phenotypic effects of that. Lots of words there. Follow a long introduction, a long abstract, but let's get into the figures here, or maybe a little bit more background. So we're looking at the sugar glider here, which is one of several, many actually, maybe 15 or more different species of gliding marsupials. This is an adult sugar glider extending the patagium, which is the skin between the forelimb and the hind limb. You have skin called the lateral skin between your arm and your leg, and but it's not developed so that you could fly. But these mammals, they're marsupial mammals, have developed this skin into a membrane that is suitable for flying. It's interesting to consider how that membrane in a sugar glider is different than the membrane in a bat. The bat has the hand wing. There's membranes that connect all the digits, as well as the patagium, which would be connecting the forearm to the hind limb. So it's different. There's more bones in it. In the sugar glider, the hands are still functional. They could still eat normally, I guess you could say, whereas the bat would have trouble. They're comparing a lot of gliding species with non-gliding species, like the striped possum, which is the closest relative to the sugar glider. And they sequenced the genomes of these two organisms and other pairs of organisms, and other, just getting more data, more non-gliders and more glider species, to find the genes that were upregulated in glider species. And then they do their far out biotech in-pouch transgenic transformation techniques to upregulate that gene, EMX2, and see how it affects it. So they do a lot of work with very young joeys. This is a 5-day old joey. So marsupial development, they're born and they're not in the uterus, and they have to crawl up to the pouch. And I even was talking to our mammologist today, like, how is it possible that this little guy, he's 5 days old, size of a grain of rice, that bar there, that white bar is 1 millimeter, can actually crawl up to the pouch. And the female apparently licks the fur and kind of makes a runway. But it's still almost inconceivable that this little joey could actually move that far. But apparently it does. It gets up into the pouch and finds a teat, and then that continues their development outside of it's not outside of the uterus, I guess. It's not the womb in the case of marsupials, but I'm a botanist, so I'm going to continue here. They sampled DNA from the Patagium at a very early embryonic stage. From the 5-day-old stage, they did harvest material in that area. You see it on this cartoon from the paper, figure 1B. They also compared that area of skin to other areas like the dorsal region and other areas to find genes that were specifically expressed in the Patagium. So for a long time, I'm like, how the heck did they get approval to do this on these small mammals? And then I was searching around on the internet, and wow, there's a whole community of sugar glider hobbyists, enthusiasts, who raise these animals as a hobby in the pet trade, and there's lots of information out there. So that led me to realize that these are not necessarily endangered species, and even in the United States, they're restricted, I think. I mean, I didn't go so deep on the Wikipedia Google search, but they could become an invasive species, literally. So they seem to be doing pretty well, and they can rear these in the lab, and they have plenty of material to work with, and they're not an endangered species. So that helped me understand how they could actually do this work. So they used two methods to look at the DNA in this skin to see what genes could actually be transcriptionally active. So it has to be accessible. They used two methods for transcriptional accessibility in the Patagium. Here's some more images of these developing joeys. This is at 8 days, P8. P stands for postnatal. This is 8 days postnatal, post-birth. This is 12 days. At this point, I could start to understand how they could get up to the pouch, but apparently after 12 days, that's a long time to be just on your own as a little joey. I think they have to go like on day 1 up there. Somehow they're able to move. So the two techniques that they use to look for transcriptional accessibility or to find transcriptionally active genes are ChIP-seq and ATAC-seq. I'm not an expert in this. There's a genomic assembly on the top. These genes are in red and blue depending on the direction of transcription. One of my students pointed out that helped me. What I don't understand is, Ricardo or Jorge, maybe you can fill me in by email, why there's that gap, like in the genomic assembly, the red, blue, red, blue, red, blue, but then there's a blank area. Why do you even have that blank area? Why not just mush them all together? What is that blank area? If it's the intergenic DNA, I would expect that there would be so much more of that, because the human genome, only about 5% of them, 5% or much less, are the exons of genes. There'd be so much more intergenic DNA. Why those gaps are so small, I don't know. How long this particular sequence is, the genomic assembly, is this the whole genome? Question mark, or is it just a part of the genome? I'm not quite sure. I didn't have full time to go fully in depth in the paper. It probably says. But for our broad brush look at these techniques, it's important to know that sometimes they were comparing the Patagium primordium to the dorsal skin area to narrow down the genes that were only transcriptionally active in the Patagium primordium. And this is not RNA, this is just DNA. Is it accessible? And I just put some arrows here showing we're looking at the arrows going up and down. The ones that are going up, that means more transcriptionally accessible. Lower down means less transcriptionally accessible. So I'd assume they're looking around those coalescing. Both two methods show increased transcriptional accessibility, and finding the cis-regulatory elements that are close by to those transcriptionally accessible regions. Those will become important. These are the two methods, ATAC-seq and ChIP-seq. They found these transcriptionally active genes by this method, or actually we could just say they found these cis-regulatory elements or CREs by this method. Both of those two methods found thousands of cis-regulatory elements or peaks, and that led to 52,000 candidate CREs or cis-regulatory elements. That'll become important soon, that number. They then mapped those 52,000 CREs or cis-regulatory elements to 17 marsupial genomes, both in gliders and non-gliders. If you look on this phylogeny, these are all marsupials with some gliding species, only 3 shown on this phylogeny. There's more than that, but they'd form monophyletic groups within those groups. You see the sugar glider down there. I asked my student what color is sugar, and they said pink, so I made sugar gliders pink here, and then the striped possum is a close relative of that sugar glider. Really trying to do some genomic comparisons of those two to find the genes that are unique in the glider species is part of this paper. But as part of this work, they sequenced 15 new genomes, so kudos for the investigators for doing all that work. I'm sure a lot of the intermediate authors did some of that sequencing, or maybe they just paid somebody to do it. I don't know how those things work these days. They had 3 glider genomes and 14 non-glider genomes, and we're just coming from studying Jay Stortz's hemoglobin paper, where he had 29 pairs of high elevation versus low elevation birds, and so my students might be a little disappointed that there was only 3 glider species here when there's more. Maybe these 3 are representative of the 3 monophyletic groups of gliders. I should have nailed that down before I did this presentation. I didn't, but I assume that must be the case. But you know, you can always criticize a paper for what they didn't do. Instead, we should more focus on what they did do, which was very impressive. So they looked for regions of DNA that showed elevated substitution in gliders. The gliders are shown in red on the phylogeny. They're calling these GARs, or glider-associated regions. These, again, are regions of DNA that show elevated substitution, but just in gliders. And these are cis-regulatory elements that show elevated levels of cis-regulatory mutation. So for each glider species, they found a number of glider-associated regions. They did a Venn diagram, and they found that none of them overlapped. That is, no single glider-associated region was expressed in all three of the glider species that they studied. So they were a bit bummed out about that, I'm sure. But the conclusion of this study is that there was one gene, EMX2, that had a glider-associated region associated with it in each of these glider species. So it's not the same glider-associated region, but EMX2 at least has at least one glider-associated region in each of these species. So the same gene is being altered in expression in all three species, but through a different mechanism, a different glider-associated cis-regulatory element. So now we're going to look at another way that they were able to narrow it down to EMX2, and that's by comparing the number of glider-associated regions to the number of cis-regulatory elements. And this is going to involve this 52,000 candidate crees as one axis, and then the glider-associated regions as the other axis. This plot becomes important. I'm not showing you the data to begin with. The x-axis is the number of GARs, or glider-associated regions, near the gene. There's going to be 1,000 points on here, and each point is a different gene. So each gene has a certain number of glider-associated regions, and then they have another axis, the y-axis, which is the total number of regions near the gene, and that was a little bit confusing. They said regions, and I'm going to say cis-regulatory elements near the gene. I think that's a safe assumption. So if we look at the category of genes that have one glider-associated region near the gene, there's some that are enriched for accelerated elements, and there's some that are enriched for accelerated elements and upregulated in the Patagium, and those are the orange dots. And then the gray dots are just not enriched at all, except they're still going up the y-axis. I don't quite understand that, and they still have a GAR. But the orange dots, focus on the orange dots. As I reveal all this data, the orange dots represent genes that are enriched for accelerated elements in gliders. They have more GARs, and then they also have more cis-regulatory elements. So EMX2 stands out as kind of a something, all indicators are pointing towards EMX2 as an important gene in this genomic pipeline, where they're really just trying to discover what are the genes. There's other genes involved here, well, at least that are showing up, but this paper ends up studying EMX2 because it has the greatest number of GARs. But we see other genes too, and it begs the question, I'm sure the reviewers are like, why didn't you do the transgenomics or in-pouch transgenics with the MAP1B and the KIRL3 and the TCF4 and the CD248? That might have led to similar results. And this is a work in progress. I'm sure somebody's following up on that, maybe, or, well, yeah, there's always going to be more to the story, I'm sure. They tried to take it to the next level of looking at the interaction between these regulatory elements, and it's difficult to explain this chart. We're looking at contact loops by this method called micro-C. We're only looking at the contact loops for EMX2. You see EMX2 on the genomic assembly up at the top. It's a gray arrow pointing to the right. There's other genes within that assembly. There are cis regulatory elements. Each of them have a number here. And using this method called micro-C, they were able to see interactions between those cis regulatory elements. And we see a major one there, 16519, is interacting with those other ones that are closer to EMX2. This is a major conclusion of their paper, is the interaction between cis regulatory elements that are producing these phenotypic effects. And they also use this crazy chart, which I'm suggesting maybe I should get this as my next science tattoo. I'm not sure I will, but it's a correlation. I think of this as a correlation plot of genomic accessibility, question mark, question mark. It's not expression, because we're looking at a genomic assembly on the x-axis on the bottom. We're looking at interactions from one point on the x-axis to another point leading up to a point, which is indicated here as EMX2, which is a darker point, indicating that those 2 points within the genomic assembly were both accessible. They may both be cis regulatory elements that are involved in expressing EMX2. Sometimes these cis regulatory elements are somewhat far away, even though they're cis regulatory. Begs the question, are they now trans regulatory? I'm not sure. But this was a kind of a cool plot to look at. I had to put this whole paper down for months because I didn't think I was going to be able to understand it well enough to present to my class, but I think I've kind of been able to be like, well, there's a crazy technique here. It's kind of like when you get to the stats section and they're talking about Bayesian Markovian chain markup analysis, and you're like, well, I guess I got to just kind of trust you on this, and it's a cool plot, so we'll just kind of leave it there. But I can understand this next technique where they did this in-pouch transgenics, where they injected a lentivirus with a vector. We could call it a vector. The virus is the vector, I guess, that leads to the expression of this short hairpin RNA. SH stands for short hairpin RNA. And this, in this case, is a short hairpin EMX2 to 3. And I don't know what the 2 to 3 means, but I'm thinking it's a short part of EMX2. Is it really 2 to 3 base pairs? I doubt it. I would think it'd be more like 6 to 7 or so. Point is, it becomes duplexed with the full transcript of EMX2, and that leads to RNA silencing through the Argonaute pathway. It leads to downregulation of EMX2, is for us maybe non-technical molecular biologist. It somehow, this lentivirus leads to downregulation, and the somehow is basically RNA interference. However, they never use the term RNA interference in the paper. Instead, it's this other technique called the short hairpin RNA technique for downregulation of a gene. You see the little Joey there in the pouch of a sugar glider. I guess that's the pouch. It's kind of hard to tell there, but maybe those are somebody's gloves, fingers, opening up the pouch, so you can see the blue in that picture are somebody's fingers. But you can see the Joey, you see its head, and then you see that red skin, looks irritated. That's where they injected the skin with the lentivirus, right there in the developing Patagium. This looks like, well, I don't know what day this one is, but it's young. Now we're going to look at, they also did a technique of injecting the lentivirus as a control with a scrambled sequence. It wasn't specific to any gene. That's just to make sure that the virus itself wasn't causing these changes in the Patagia development. They also did a control, proof of concept, I would say. They injected GFP instead of the EMX2 or with the EMX2. Maybe they always had GFP in addition to this short hairpin EMX2 RNA. And point is, they were able to transfect in the pouch. You see they did the bright field, doesn't have anything, GFP expression in the middle panel, more expression in the Patagium where they did the injection. On the other side, they did the scrambled sequence, or they didn't inject with the GFP. That shows, well, the lentivirus technique seems to be stable. It doesn't spread through the entire body. The stent tends to stay in the tissue where the injection site was. And then they show that the GFP is actually pervasive in all of the cells within a cross section. That's the right hand panel. So it is pervasive, it's everywhere in there. And then we look at the data, the Patagium area in the EMX2 short hairpin expressing is less. I'll just slow down for a second. The Patagium area was reduced in the short hairpin EMX2 injected Patagium versus the scrambled injected Patagium. So the left hand versus the right hand side. The left hand side with the scrambled is a certain size, it's the control, it's a 1. With the short hairpin EMX2 in the lentivirus, it's smaller. It's significantly smaller. And that's their whole point. They're doing RNA interference for EMX2, needs EMX2 to make the Patagium. If you don't have as much, it's less. It's not as large. Then they actually looked at EMX2 expression level using qPCR, and sure enough, there's less EMX2 in the short hairpin EMX2 lentivirus injected Patagia. A lot of words there, and to get to the point, compared to an uninjected Patagium. So that's cool. They were able to do that in pouch, transgenics or transformations. And then they also showed with immunohistochemistry that the levels of the EMX2 protein was reduced in the Patagium that were injected with this lentivirus containing the short hairpin EMX2 versus the uninjected controls. We're seeing the uninjected control up at the top, and we're actually seeing more EMX2 protein. I make a point for my students that the EMX2 all caps is referring to the protein. Here, the antibody staining reporter assay showing red for more EMX2. Then we have to kind of use our eyes to see that in the injected EMX2 with the short hairpin RNA, there's less EMX2. It seems like it's a smaller Patagium in general, but that's the point, is the EMX2 is leading to a larger Patagia. When there's less, it's smaller. We see that in these comparisons, but of course we'd like to see more data on that, and there was quantified data on that too. Then they switched to EMX2 in mice embryos. We see that EMX2 is expressed in mice embryos at embryo 11.5 days. So this is a small mouse. You see the eyes, and then you also see the Wolfarian ridge developing. EMX2 is being expressed in lots of places, but it diminishes as we get to, this is just 2 days later. It's right behind me. It's the E13.5. The Wolfarian ridge is no longer visible. We still see a little spot up there, probably still in the eyes. There's a few spots with arrows there. Point is, mice are expressing EMX2 normally, but then what happens when we overexpress EMX2 and they have a transgenic mouse that has elevated levels of EMX2 expression, and they first assayed for keratin. Keratin is a skin protein. KRT14 is a keratin protein that's in skin, and we're just looking at it here probably with an antibody stain in the transgenic mouse on the left and a littermate control, non-transgenic on the right. So they're is a little bit of EMX2 in all mice, but it's overexpressed, and that leads to more keratin, more skin cells basically, and that leads to thicker skin and greater cell density in these transgenic mice, and there's more data to support that conclusion. The epidermal thickness was larger in the EMX2 transgenic mice, that's the red bar, and the cell density per surface area was greater in the EMX2 transgenic mice as well. So just to finish up some glossary notes here, the ATAC-seq is an assay for transposase accessible chromatin. It identifies areas of chromatin being expressed or accessible to RNA polymerase. ChIP-seq, so much to go into there. This presentation, it won't do it justice, but the title ChIP is chromatin amino precipitation, and then it's followed by sequencing. We could stop there. The next part is acetylated lysine on histone 3, provides information for which of the ATAC regions correspond to active enhancers and promoters. I tried to understand what the acetylated lysine on histone 3, basically it's epigenetic modification of the histones. It has to be so that the DNA is unwound, and if there's acetylation on histone 3, that means it's either more or less accessible, something like that. Again, we just kind of have to trust them that this was a method for identifying transcriptionally active genes. A CRE is a candidate cis regulatory element. A GAR is a glider accelerated region, and shRNA is a short hairpin RNA. And I'm just saying it's a method for RNAi, even though, well, that's what that says on the internet, and I hope that's a safe assumption, or at least in general, that's what's happening. A way of knocking down gene expression. All right, I'm going to go back to my take-home points. Sugar glider Patagium have evolved repeatedly in marsupials. It's a convergence within the convergent evolution textbook example. Transcriptionally active genes in the Patagium were discovered and compared to non-glider and glider species or genomes to identify glider-associated genes with elevated substitution in the cis-regulatory elements. That's what pointed them to the genes, I'm going to say genes, even though they identify EMX2. They found other genes too, but they kind of leave those in the background, at least for my story here. EMX2 stood out in their transcriptomic epigenomic genomic pipeline, and in-pouch transformed joeys and transgenic mice with increased or decreased EMX2 expression had altered Patagium primordia. And gliders show lineage-specific regulatory evolution. Incredible work, Jorge and Ricardo. Thank all your investigators, and way to go. Looking forward to seeing the next installment of the non-model system evo-devo. Thank you.